So thank you for coming. My name is Fabian Michelangeli. I'm a curator here in the Institute of Systematic Botany. Some of you know me, but some of you don't. I've been here at the garden for 16 years now, and I do my research all over the neotropics. But today, I will be emphasizing not only my work, but the work of the garden in general in Brazil. So there is a lot of, when I say I, and I'm talking about me, is really the work done by the students, the collaborators, and everybody else. So, Brazil is a big country. It's large, and it has over 33,000 species of flowering plants. To give you an idea, the US plus Canada has less than 24,000 plants, and in a territory that is 4.5 or 5 times larger than Brazil. So fewer plants in 5 times. And in Brazil, we can divide the biome roughly in 6 distinct areas. The Amazon forest, if we start from the northwest, the Amazon forest, the Cerrado in yellow, the Caatinga in brown, the Atlantic rainforest in light yellow, and then the 2 smaller ones over there in the west is the Pantanal, which is the flooded grasslands, and the Pampa, sort of the plains in the south. So in total, about 55% of the plant species in Brazil are endemic to Brazil. And those 33,300 species represent over 15% of the known number of flowering plants on Earth. But maybe the actual number in Brazil is over 40,000. And I will talk a little bit more about that in a bit. So just to give you some images of how this looks. So this is the Amazon forest. This is, 1.3 billion acres, and just the Brazilian part. It's not counting the Amazonian forest that extends into Bolivia, Peru, Ecuador, Brazil, Colombia, the Guyanas, Venezuela. And there are about 14,000 species of plants in Amazonia, and about half of those are trees. And sometimes it's very easy to collect things that are about this high, but when the trees are 40, 50 meters tall, we don't have as many plant collections of those trees. So that's a lot where our knowledge vacuums are. And not only the trees, but all the things that grow onto the trees, we know even less about those epiphytes up there. So even though we seem to know a lot, we have collected lots of plants, there's still more to find. Then the Cerrado, the grasslands of central Brazil, it's about 25% of the country, and it has over 10,000 different plant species. If you think of grasslands, we tend to think of the plains in the Midwest, and there are usually not as many trees, but the Cerrado in Brazil is like a quilt of different types of geological formations and rivers that cross it. So then it's not a flat grassland, but it has lots of areas with trees, whether it's in escarpments, along the rivers and so forth. And within the Cerrado, there are special areas, and they are one of my favorites, is the Campos Rupestres, which are these high altitude rocky fields. So areas above 3,000 feet that are distributed within the Cerrado. And the Campo Rupestre is a little bit of an extreme environment because the temperatures can go very low into the 30s or 40s at night, but they can be in the hundreds just 12 hours later during the day. So you get moisture that condensates during the morning, and then super hot and dry during the day. So the plants in the Cerrado can experience in 24 hours the cycles that a plant in New York would experience in 1 year. So they're very interesting adaptations, and it's a very beautiful area. Then there's the Atlantic forest. And when we say the Atlantic forest, you look at it on a map and you think, oh, well, it's just the coast of Brazil. But because we're not used to the dimensions and the scales, we forget how big that is. And the Atlantic rainforest, if you start in the state of Pernambuco and you drive all the way down to the state of Rio Grande do Sul, that's all Atlantic rainforest. And it will be the same distance as if you went from Portland, Maine, all the way to Key West. So and anybody who has driven or even flown that, you know that the plants are very different in Maine than they are in the Everglades. So the same thing happens in the Cerrado. In the Atlantic rainforest, where we have a big diversity of habitats. So in the central part of the Atlantic rainforest, we have a lot of this Inselbergs, these big granite mountains. And then towards the south, we have the Araucaria forest, and then we have more lowland forest towards the north. So there is, as you can see, a lot of variation there. So the garden has been working in Brazil really even before the garden was the garden. Expeditions by Rusby and others to South America from the 1860s to the 1890s really created the first collections that Nathaniel Lord Britton was trying to put names to. And then those were the collections that when he went to London, and so Kew Gardens inspired him to come back and lobby to create the garden. So when we can say that the garden has been working in the Amazon before the garden was the garden. And it's really instrumental. So there we have first expedition, 1869, something like that, by Rusby. That continued. I think that might be Wade's picture in the late 70s, early 80s. And then more recently, we're still working in all areas of Brazil. So this is a continuous work in the country. And, when Rusby went, he just collected the plants. Actually, Rusby was a little bit different than most. He also did a lot of economic botany collections and things. But in general, most of the collections we did for the first 100 years were the plants. But as things have evolved, the collections have also evolved. And when we go down, we now are taking photographs, we're collecting samples for DNA sequences, we're collecting samples to do anatomy, to do electron microscopy, we're collecting all sorts of things that we don't even know exactly how they might be used. In fact, within the life of a project, I collected a lot of samples that were supposed to be for DNA sequencing, and then we realized that the ways they're being preserved for DNA sequencing was also the best way to preserve them to do electron microscopy of the surface of the leaf. So the sample that was collected for one purpose, within 5 years, was being used for another purpose. And that's a very important aspect of building collections, is that you collect as much information as you can, and you preserve it and curate it as well as you can, because you never know what is the impact of that collection down the road. Sometimes 100 years, sometimes 5 years. So through all of that, another thing that has changed significantly is that, when exploration started, it was very, let's say, imperialistic. We went down there, we collected, we bring things back. But now, collections are really done in true partnerships with our Brazilian colleagues if we're in Brazil, Colombian colleagues in Colombia, Peruvian colleagues, etc. And in fact, so this is one of my own collections from Rio de Janeiro, but you see that the label is from a university in southern Brazil, and it's because they are our partners there. They are the people who are in charge of the logistics and everything. And the other thing that you see here is that we have very precise localities. In the past, if you read Rusby's collections or Spruce's collections, they might say Brazil or Amazonas. Well, that doesn't help a lot. But when you get a lot of precise collections, you can start to put them in a map. And the garden has over 400, I think the number is more like 430,000 specimens for Brazil. And when you put them in a map, it looks like there is a lot. But I will argue that is very, very little. And we'll talk about that in a minute. One of the things I want you to look at is, look at the Amazon, which is that part over there on the upper left. And it's not as dense as the Atlantic forest. And then you see those straight lines there. And those lines are, for the most part, rivers. So we are collecting only along rivers. The rivers are the highways of the Amazon. All the transport happens along rivers. You even have a city like Manaus that has 1.5 million people that is not connected by a real road to the rest of Brazil. Everything gets to Belém and then goes up by river. So the rivers are the highways there. And you see, so the areas in between the rivers, we lack a lot of information. So one of the things about Brazil is that it has a lot of forests. 56% of the country used to be covered by forest. But things are changing very quickly, or have been changing actually for the past 500 years. And there are lots of different pressures on the forests of Brazil. One obvious one is urbanization. People arrived and they needed land. So here we have Sao Paulo, a city of 20 million people, right in the center of the Atlantic rainforest. Clear land for cattle ranching. And even you can go online and see, oh, this looks like a nice forest. It's a beautiful forest. And then when you look at it, every single one of those things is an eucalyptus. And that really doesn't, it is forest, but it's not native forest. There's no diversity. There's one species there. So it might look from Google Earth as you have a lot of diversity, but we've been clearing the natural forest to plant things for wood, for paper, for construction, etc. And nowhere this is more dramatic than in the Atlantic rainforest. So everything you see in green in the map in the middle there is what the Atlantic rainforest was when the Portuguese arrived in Brazil in the early 1500s. And this, what you see here, is what was left in 2008. So there is less than 8% of the forest, of the original surface, is what remains. And even more dramatic, most of the patches of forest that remains are less than 1,500 acres each. So if you have such little patches, now instead of having one continuous rainforest, Atlantic rainforest, what you have is something that looks more like an archipelago of forest. So they are all little mini islands. So there's over 150 million people that live within the Atlantic rainforest domain, or what was the Atlantic rainforest domain. It's the 8th largest economy in the world. So we need to take action. And for the past 6 years, we've been working on a project that was funded by the National Science Foundation, NASA, and the FAPESP, which is the equivalent of the National Science Foundation, but for the state of Sao Paulo in Brazil. And a project that is, has coordination from the Botanical Garden, CUNY, and the University in Sao Paulo, with all these other institutions, both in Brazil and also even in Australia and in France. We've been trying to find out what are the processes that drive biodiversity and that can help us predict what will happen to biodiversity in a changing world. So this is a group of over 50 researchers, students, postdocs, et cetera. And we have climate people, we have botanists, we have zoologists, we have palynologists, so people that look at the pollen that has been deposited 1 million years ago. So we have a very big team. So one of the things is that if you see a plant here and you see another plant, let's say in Manhattan, but there is no forest in the South Bronx, you cannot tell if the plant was there or not, or can you? So what we do is we use those dots from the herbarium, from the distribution of the species, to try to make models. So I have here an example of one species that is a very common one that we have lots of collections of. And then we take information from the rainfall, the elevation, the soil moisture, the temperature. And this can be information that comes from remote sensing, so this is where NASA comes in, or from models. And then we create a map trying to understand whether a plant is present in a place or not. So if we see something like this gap here, is this gap here because the plant was never there or because we destroyed the forest? And same for this like little gap here. See, I touch the screen and it's gonna get crazy. So here is the map. So everything that looks orange is where the plant, high probability of you finding it there. Everything in yellow, medium probability, and blue, no, low probability. So you can see here, this area here is in red. And that is the gap over there. So that area should have had that species, but it doesn't have it anymore. But here we have these areas in blues, which are also a little gap over there. Those seem to be real absences. So once we do that with one species, we can create individual maps for many, 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 many species. So each of those 6 maps over there on the left are individual models. So we took, in some cases we have 10 points of distribution, sometimes we have 300 points of distribution, sometimes we have 8, hopefully more than that. And then we create all those maps. And you can see that there are species that are widespread. You can see species that are on the coast. You'll see species on the mountains, species that are microendemic. And then you sandwich all of those on top of each other. And you can end up with a map of what we call in biodiversity research richness, just meaning the number of species in a place. So the warmer the color, the more species it is. There are present. So there are more species here than there are here in blue. So that map is only the map for the group that I work on, the Melastomes. But then we have other information. So we not only know where they are, but we have DNA information, and we know how plants are related to each other. So we can incorporate that information with some mathematical formulas, and we can see that the map in the middle now takes into account how things are related to each other. And there are areas, for example, here and here, that are not so hot color here, that are hot color here. And it's because there are not as many species here, but the species there are all of different lineages. They are not close cousins. So if you are trying to think of how to preserve species, yes, there are fewer species here than here, but maybe the species here represent a larger sample of the genetic diversity than the species in other areas. Or we can just decide where are the species that are microendemic, the species that are only found, let's say, in patches that are less than 1 mile by 1 mile, which is what this map is. And because the patches are so small, you cannot see it, but there are some tiny red and yellow areas. Oops, let's go back. Tiny red and yellow areas here. So these are some mountain summits that have incredible amount of endemic species. So, or what we call microendemic. So there are many ways that you can start to look at the same information, but depending on how you ask the question. So this is only the Melastomes, the group that I work with, and not even all the Melastomes in Brazil, which there are about 1,300 species. This is one particular group of Melastomes. So I said that our team had lots of different people, and don't get dizzy by all the colors. What is important here is that we have maps that we are doing the same procedure with bromeliads, the Melastomes, a group of butterflies, a group of toads, a group of frogs, 2 different groups of birds, and we can start to layer all of that information together. And we have chosen those groups because those are the groups where we have specialists in. We're not going to choose a group that we don't have any genetic information, because I cannot do genetic things. We cannot choose a group that is very, very rarely collected where I only have 1 specimen, because then I cannot do the models. So there are a lot of requirements to try to understand and how the climate and the species go together. But once we put all of that, we can create a map, which is the map all the way on the left there, about how communities are different. So now here, if the color looks similar, it's the same community. If the color looks different, it's a different community. So you remember how I said the Atlantic rainforest changes, from Portland, Oregon to Key West, in this case from Pernambuco to Rio Grande do Sul. And you can see the color here is different from the color here. And this is not an immediate change. It's a gradual change as we go down. This is a completely different community, etc. And then we can do the same. We can do like a multi-dimensional map of the weather, the climate on that same area. And we see that the transitions tend to happen in the same places, but not in the exact same places. And it's because not only climate is important for the distribution of the species, geology is important for the distribution of the species, and something that we cannot easily capture is the interaction between species. If you are an orchid that can grow everywhere, but your very specialized pollinator can only live in this one mountain, then you're gonna be restricted to that one mountain, and that we cannot capture with this climate models yet. But what is interesting is that one of the things we're learning here is that not all plants, we can separate it from groups or animals, groups of plants, not all organisms are responding the same way. So not surprising for frogs and lizards that they're not warm-blooded, they need to go out in the sun to regulate the temperature or get in the shade to regulate the temperature. Temperature is very important to where they are found. But for some plants, temperature is really not important at all, but is rainfall. And not only rainfall in absolute numbers, but whether it rains in 2 seasons or where the rain happens more evenly throughout the year. So we're starting to understand what are the factors that are important for each of those species. And we have thousands of species in these models now. So the maps over there on the left tells us how much is left of each of those environments within the Atlantic rainforest. And you can see the little green sort of peninsula going there on the upper left. Only less than 1%, 0.6% of the native vegetation is left in that area of Brazil. So now we have here the number of species. Let's look at the one in the middle, or the phylogenetic diversity in the middle. If we have not touched the forest, but because we have destroyed so much of the forest, there's nothing left here. I mean, there's things left there, but the dots are so small, you cannot, they're smaller than a pixel on the monitor here. There are patches of forest there, but they're tiny. So those species there obviously are in more trouble than the ones in this area around Rio de Janeiro and Sao Paulo. So now we start to think, okay, what's going to happen to these species? And we can start to use models to see how temperature, soil moisture, seasonality of the rain, et cetera, is going to change moving forward 50 years, 30 years. So in this case, this map is for 2050, so 30 years. And you can see it's not that it's going to get warmer everywhere. It's not that it's going to get drier everywhere. So most places get warmer, but some get colder. Some get like the changes in temperature are more extreme. So if you see the one in the top left, that's annual precipitation. Actually, in western Amazonia, there's going to be more rain. But in eastern Brazil, there's going to be less rain. That's what the models predict. So it's not like drought everywhere, or it's not floods everywhere. It's different places react differently. So then when we take that and then we take the models of the species, we can try to estimate where they have to go. So each of those arrows there represents a group of species in just one study. And then so the species that were like here, they have to move north. This species has to move this way. So not every species is moving in the same direction. Not all species are going south to counteract temperature, or uphill to counteract temperature, because not all depend on temperature. So you see the species are moving in many directions. But where they need to move might not be available anymore, because we already chopped down the forest. And if we calculate that for the different target groups, we see that groups are affected differently. So in the tall bars in sort of bluish are how much their range would change if there was no deforestation. And the one in gray, the much smaller one, says how much the range might change if there is deforestation. And a group like those bromeliads, that particular group in the pineapple family over there on the left, you see there's very little. They're gonna have very little to go to. But it's because they are already at the top of mountains, isolated areas in the state of Espiritu Santo and southern Bahia. They really have nowhere to go. The birds, yeah, it's gonna be affected, but they have some places to go. The other thing is, it might be easier for a bird to get to another patch of forest that is for some species that have a big seed like this or something, So, and those things are very hard to take into account. Sorry. So, originally, when we planned this talk, I was going to talk more about the Atlantic rainforest. But we've been reading the news, and we know that the Amazon, it's been very prominently displayed due to all these fires. Well, unfortunately, this is not a thing that started happening this year and didn't happen last year. Fires have been happening in the Amazon for a long time. And here we have all the forest has been lost in Brazil since 1985. You might notice here that there is very little forest lost in eastern Brazil. Well, that's because that was chopped the previous 450 years. It's not that they're doing that well there. But southern Amazonia is the one area that's been really, really affected in the last 35 years. And there is both demand for wood, but also for more land to plant corn, sorghum, maize, all sorts of different things. So here we have a different version of the deforestation, just looking at Amazonia. And you can see that everything that is in green or tan is a forest preserve. And you can see how those forest preserves are somehow helping. So you have this, like for example here, this very big area of no deforestation because that's a forest preserve or a national park or a native indigenous community land preserve. So they are having an effect, but even within some areas that are preserves, deforestation is happening. So it's not the magic cure. So the typical way this happens is people get assigned a parcel of land. Doesn't have to be a huge corporation. It can be a small landowner, or it can be a corporation that's going to plant sorghum. And they have to clear the forest, but then there's a lot of debris behind that they cannot haul away. All the leaves, all the trunks they can sell as wood, but all the leaves, the branches, all the shrub that was underneath, they can haul away. So they wait till it dries and then they put fire to it. And you can see that on the photo on the left. And look how close that is to the forest. So not only is burning the actual forest, they're not burning the area that they have clear, but those fires also get into the forest. And you might go with a satellite image and still see a forest there, but what you are missing is how those fires have burned the leaf litter underneath. And that's one of the problems that this current set of fires has really shown, is how the fires go into the forest. Doesn't seem that it affected the forest, but it's effectively killing it, because the nutrients are very quickly recycled in the Amazon rainforest. And if you kill the leaf litter, you basically are starving the trees from all the nutrients. So in the map, in yellow, we have everything that has been cleared in the last 35 years. Is huge amounts of area, and I will give you some perspective in a minute. And everything that is that sort of tannish, brownish color is fires that were active during the month of August this year. So it's not that it's everywhere in the Amazon. The fires are exactly at the frontier of the people moving in the Amazon, where people are that's where fires are happening. Is it worse this year than last year? Yes, it is. Is it worse than it was in the 70s? Maybe yes, maybe no. Part of the problem is that we weren't taking the same types of satellite images in 1971, which was a very, very dry year, than we are doing now. So it's hard to compare sometimes these numbers, but there are definitely lots of fires. So one of the areas where we see the most deforestation and the most activity is here in the state of Rondonia. In the state of Rondonia, it's about the size of Oregon, okay? And it's everything from that yellow line on the left to this white line here on the right. That's the state of Rondonia. So the size of Oregon, and look at how that forest has been cut away. And 100% of that has been lost in the last 35 years. The area cut there in the last 35 years is bigger than the South Carolina in 35 years. And you see there are some areas here in the center and then towards the north and towards the east of Rondonia where there are still good patches of forest. And one of these areas is and then we go and look in our herbarium. How many specimens we have for the state of Rondonia? What do we know about the state of Rondonia? Well, even though this is one of the areas where humans are most actively working, we only have 10,000 specimens for the entire state of Rondonia. It's not much. It's one of the most under-collected areas of the Amazon. And not only that, look at where the collections are, which are all those red points. They're basically in the areas that have already been cut down, where there are roads where people have access to. Or, in this case, they went along the river, and even these habitats don't exist anymore because they created a dam, and so those plants are gone too. So we need to take action, and in fact, our colleague Doug Daly, who would have been probably a person much better qualified than myself to give this talk, is not here because he is in Rondonia right now. He left last Friday, and Doug is working right in the Floresta Nacional da Jacunda. That's where he started to work about a year ago. And that national forest in that area in northern Rondonia has one locality that has been collected, and that is before Doug started there, and that's where the forest manager lives. And this is a national forest. This is not a national park. So this area can be exploited. This area can be extracted, can be used. So it's very critical that we understand what's happening there. I encourage you to go to Twitter, Facebook, Instagram of the garden. And Doug has posted 2 small videos already, one 2 days ago and one this morning or yesterday about his trip there. I suspect we might not see one for the next couple of days because there's no internet in Jacunda, but once he gets back to Portobello, he will be updating us on the work they're doing there right now. So, as I said, 430,000 specimens collected in Amazonia, but there's still lots of holes. And there are, of the 12,000 species in Brazil and Amazonia, we have specimens of over 90% of them. I mean, that is pretty impressive. But 4,000 species, 3,900 species, we only are represented by a single specimen. And 8,200 species are represented by less than 5 specimens. So we really cannot understand a lot of what's happening with that few collections. In fact, most Amazonian species are only represented by 12 specimens. That's the average. 12 specimens per Amazonian species. That is 1 specimen every 177,000 square miles. That is the size of California. In fact, that's bigger than California. So if you had 1 collection from San Diego, what could you say about the distribution of that plant, about the biology of that plant, about the conservation of that plant, about how vulnerable that plant is? So 1 specimen for the size of California is the average. I'm going to show you one that has twice the average. This is a species in the bird of paradise. It's a Heliconia. So this has 24 specimens in our herbarium. And look how far apart those dots are. So is this species found here in northwest Amazonia? It seems that it isn't. Well, I went to another herbarium database, and they have like 3 dots there. So unfortunately nowadays, as more and more herbaria get digitized, we can share that information with our colleagues. But it's still insufficient. In fact, if we add the things that I looked at, which was a herbarium in Manaus, we went from 25 points to like 32 points. Because 20 of our 25 points are also in Manaus. So it's not that if you add their collections and ours, it's not adding one and the other, because there are duplicates everywhere. So what I really think is that the garden, what does the garden have to do? We have to do what we've been doing for the past 130 years. We really have to keep doing what we have been doing. We have to keep collecting. Now, we're doing a lot of things that we weren't doing before. So not only is getting out there to areas that nobody wants to go to because you have to hike 3 days to get, but for example, here in the lower left is our colleague Ben Torque in Tapajós, also part of southern Amazonia, another area that is lots of unknowns. And in fact, Ben was able to get funding from the National Science Foundation to collect in this area precisely because there was no information. And Ben is not alone. And Ben is not only with colleagues there on the left, but Ben is with a lot of local students. So he is creating or helping form the next generation of botanists in Brazil. That's very academic. So here we have Doug in Rondonia working with local woodsmen. So as I said before, the forest, one of the places where he's working, they actually use this forest. But they don't know the difference between common species that you can exploit somehow sustainably to the very rare thing that nobody knows if you're gonna just get it extinct by cutting the one tree that is every 500 acres. So Doug, instead of just saying, let's stop everything we're doing, taking a more pragmatic approach to train the people that are metaphorically and literally boots on the ground, helping manage this forest. So they can identify the rare from the common species, the economically sustainable ones from the ones that are not. So that's the work we've been doing, but we just have to do more. And you know, when I say that we have an area that is almost as big as the continental US, and there's only a handful of botanists, it is a big chore. So I want all of you guys to help us to keep working here. Now, not everything is is necessarily bad. Here we have, this is not the Itatiaia National Park in literally halfway between Rio and Sao Paulo. This is the oldest national park in Brazil. And even though it's in an area, if you count the populations of Rio and Sao Paulo, and this is smack in the middle, you have almost 50 million people in those two states. The national park is still there and is thriving and is beautiful. And so conservation can work. Habitats. It's not about conserving one species, it's about preserving habitats and knowing the habitats. So I was here just 6 weeks ago. We found a plant that had not been collected in 130 years. And it's not that I would lie to you if I told you I had to hike like a madman to find it. I found it on the side of the road. But the difference is that I was looking for it. Nobody had gone looking for this plant, probably in the last 130 years. Nobody had seen it in the herbarium and say, oh, this thing is so rare, let's go find it. Well, I went and I found it because it was on the side of the road. But not far from this, there's another area, the Parque Nacional da Bocaina, where we also went during this same trip. And in one day, we found 2 new species. And I know they are new because they are in the groups that I work with. I only know 0.1% of the flora. I don't know the sedges that Wade has been collecting. He always tells me to collect them, but I don't, because I don't can't tell them apart. So I don't know, if I collect one, I don't know if the one that I see 10 meters later is the same or is another one. So what is important here is to keep collecting and keep looking at the biodiversity. And it's the only way we can preserve. You cannot preserve what you don't know. You cannot fence it. If we could fence it, that would be a solution, but we cannot do that. So we need to understand what is there to move forward. Thank you. Questions?
How did you do the phylogenetic diversity modeling in the past? Is that based on their variant specimens or?
No, so no, there were no maps for the past. So each individual species gets a model, okay? And then we can calculate, we have a tree, and we can calculate the distance from species 1 to species 3. So for example, you know, the acai palm is more closely related to the coconut than either one of them to guarana. So then for each point, we can add the number of species that are there, taking into consideration those distances. And then we use the climatic models to go both back to the past and to the future. In some cases, we have pollen information. So I have a project which I didn't talk about here on Araucaria. It's the monkey puzzle tree. It's a perfect model to do this thing to the past with pollen because there's only one species of Araucaria in Brazil, and the pollen is very distinct, very different. So if you see that you make a hole down hundreds and hundreds of meters, and then you slice it, you put it in a microscope slide, and you count, you can tell whether Araucaria was present or absent in a given place. So one of our colleagues that works in France, she has cores all over Brazil. So we know areas in the state of Minas Gerais had Araucaria only 6,000 years ago, and now there isn't there. But we cannot do that for all the species because not all the species have pollen that is preserved, in most cases, you can only get to like family. Oh, this is the pollen of a sedge, this is a pollen of a grass, but you cannot tell which grass or which sedge.
Hi, this is fascinating and horrifying and with so many different animals and gods, or maybe that's just me. I'm curious, where you have this massive, incredible collaboration between various groups of people, and you've got this incredible set of data. And so I was left wondering, you know, what is sort of the objective for conservation policy of your particular project? So what I'm thinking about is like the EDGE program that is sort of being put in place, particularly thinking about phylogenetics.
Yeah, so it's the impacts to policy are complicated because it's hard for us from here in the US to tell the Brazilian government what to do. But fortunately, we have Brazilian colleagues that are actually pushing that. And sometimes they push it at a state level, sometimes they put it at a municipal level, sometimes at a national level. Also, Wade has a lot of experience with this. You might not be able to convince the government to do something, but you might be able to convince private donors to start preserving certain areas. And then, unfortunately, the politics in Brazil, I don't want to necessarily talk about the politicians, but the politics in Brazil, like right now, are not very open to what I have to say. So, coming from abroad. But that's the good thing is that we have funding from a Brazilian entity, and we have colleagues in Brazil. So it is a complicated process, but you cannot even do the policy if you don't have at least the basic knowledge. I have a little bit of a funny accent, I know, but I'm not Brazilian. I'm Venezuelan. I'm from Venezuela, just to clarify that.
Have you benefited from citizen science initiatives such as iNaturalist to fill in those gaps? So, I know there's a weasel that someone saw in a bathroom in Colombia and they thought it was extinctive.
Yeah, so the citizen science part works in different levels. It's been already well documented that it's been helpful to find new species. People take pictures of plants, post them on Facebook, iNaturalist, Instagram, and then a specialist sees and says, that's weird, and contacts and stuff. And in fact, in the family that I work on, the group that I work on, we have, I think, 7 species that have been described in the last 10 years for Brazil, 5 for Brazil, 1 for Peru, and 1 for Colombia that were found on Facebook or iNaturalist. That said, iNaturalist is not well curated. So they put a name of a plant, and then somebody comes and puts the name. Yeah, it says, oh yeah, I agree, and that's not necessarily true. I described a species, and then somebody put a picture of the type. So that's the name of the plant, and I went and put the name of the plant because I know the person who put on iNaturalist, and I put this is the plant. And then somebody says, oh no, that's not that, and put the name of a thing in a different genus. And then you got like 3 agreements. So I went back and I said, I described this plant, I know what it is, that's the type. And then 2 days later, it was back to the, okay, whatever. So to use iNaturalist for plants is very complicated because the curation. Even for birds, so iNaturalist has a AI, artificial intelligence program with Google, and they're looking at bird images, and they can determine, based on field pictures of birds, correctly about like 97% of the birds or something like that. Well, that's great, but that, there are a lot fewer birds, the photographs are a lot better, and a lot more photographs to train your algorithms with. We don't have that with plants yet, although we have a project where we're looking at herbarium specimens to do identifications with these type of algorithms, but yeah. And then other citizen science, I think, where it has to go is more towards the conservation of the environment, of places.
I want to add that we also have citizen scientists helping us collect data from herbarium specimens.
Correct.
So we have a Melastoma Tasty project ongoing. They're almost complete. They're about to contribute about 6,000 new specimen data entries for your group.
Yeah, so what happens is, yes, we digitize the specimens, but all the new ones, when we upload them, we are already also writing all the information label on a database, so it all goes smoothly in. But anything that was collected 50, 60, 100 years ago, maybe even 20 years ago, that information didn't go in automatically. So Charlie has coordinated a project where we get all the images where the information is not in the database, and people literally sit there to read handwriting and decipher, and then not only to read the handwriting, but sometimes then you go a step beyond that. They didn't have a GPS in 1960, but it does say 67 kilometers north by road of this place, and then you can go and find a spot and then map that and add it. And that's like secondary georeferencing, and we do a lot of that.
Fabian, could you take us through the steps? What happened when you saw that plant, which hadn't been recorded for 150 years in the park? What do? You obviously don't bend down and take it out of the soil.
Obviously, yes. Sorry, yes.
But then all of it is gone. So what happens?
So, well, first, in this particular case, it was a common plant. It was very common. So that was funny enough. This is like, well, I mean, obviously people have been coming here, but it's just been ignoring this plant. And I photograph it. I take samples for DNA. I take samples for microscopy. I take samples for anatomy. I take a precise GPS location. And then I try to get a sense of how many individuals are there. If you worked on, let's say, orchids or bromeliads, very often when you collect a plant, you are collecting the whole individual. But when you work on, let's say, something like a grass or a sedge or a rush, you have a field of these things, and then you collect a few of the individuals, and there are others that you leave behind. And then when you have a tree, you're basically like just pruning the tree. You're not gonna, fall down with a chainsaw, the whole thing. Oh, some people do, but I don't. You find something that is rare, you're not gonna take the entire population and the entire individual.
And where is this piece now? Where is your specimen now?
The specimens, and we made 6 or 7 duplicates, are still all in Universidad Federal de Paraná. And then as soon as we get an export permit, we'll get 2 or 3 duplicates up to the US. And then we'll probably send 1 to Missouri, 1 to Kew, something like that. So now we have more people, more scientists have access to these samples because they are distributed throughout the entire world. So we tried, like the one we found that I knew was new on the spot, it was a huge tree. We made 12 duplicates because we wanted to send to as many herbaria as possible. And our permit specified that the most we could do was 12. Yeah, so.
Thank you.
And are they collecting locally as well?
Yeah, so some those pictures on those are my students there or my collaborators there. So yes, it's all done locally, processed locally.
And preserved and kept there?
Yeah, so there are lots of herbaria in Brazil. So we work with many different institutions, and different scientists here work with different institutions. So as the garden as a whole probably has a network of 20 or 30 different herbaria in Brazil that we actively collaborate with. So because of that, we end up with sometimes a better collection than anybody else in Brazil, because the herbarium in the very north of Pernambuco might not send a specimen to the south to Rio Grande do Sul, but will send one to us. So, and this is true for Brazil, it's true for Cuba, it's true for a few countries where, the Caribbean in general, we have better collections than sometimes locally because we've been able to collaborate with so many people.
How do you get plants through customs?
Well, it's not that I travel with them in my backpack. I have, but you need a collecting permit from the country, and if you are inside a national park, that's another layer, and then if you are in an indigenous area, that's yet another layer that even requires the Minister of Education of Brazil, or and you need to have an actual permit from the indigenous communities as well. So once you have all of that and you have brought the specimens to the appropriate partner herbarium, you can request an export permit. And then with the export permit, you can bring them into the US. If there are any samples that have soil, that have, or different things, you may need to have an import permit into the US. And then if you have collections of things that are traded, like by amateurs, by collectors, like orchids or cycads or bromeliads, then you need another permit that is called a CITES permit. So it's some paperwork. So some of my samples, I have a colleague from Peru back there sitting, some of my samples I collected in Peru in 2017 are still in his office because the process sometimes is slow. And we have to respect that those countries are letting us work there, and we have to work under the rules. So sometimes you have to wait 5 years to get your samples out.